Well, a University of Wisconsin Superior biology professor is using tiny tropical fish in a research project that could have a big impact. Using zebrafish found in just about any pet store, they are studying the role two genes play in a type of pediatric muscle cancer. The project was just awarded a 3-year grant from the National Institutes of Health. And so here to tell us more is Dr. Jenean O'Brien, an assistant professor of biology at UW Superior. Kim Kobar is a recent UWS graduate who works as a research assistant on the project, and Vittorio Bomtempi is a student working on the zebrafish research project. I want to thank all three of you for being here. Now, doctor, obviously this is a devastating pediatric muscle cancer. Can you tell us about the research that's being done at UW Superior with you and the others there?
Yeah, so as you mentioned, we're working on two particular genes. So genes are segments of DNA that code for the functional units inside of our cells. And so we're looking at two different genes that previous patient data from some of the pediatric patients have suggested might play a role in this cancer. And we're really just trying to figure out what exactly these two genes, well, the gene products, are actually doing in the tumor cells. We think that one of them might actually be promoting the tumor cells to be more aggressive, so to potentially be able to divide more and metastasize or spread faster. And we think the other one might actually be protective, because we see the levels of the other gene go down in the tumors compared to normal tissue.
At what age is it discovered that children have this cancer, and what's the survival rate? I understand it's not real high.
Yeah, so there's an age range anywhere from under 1 year of age up and through the teens. There actually are some adults who have rhabdomyosarcoma as well. It's just more common in a pediatric subset. The survival rates, they range depending on the study, but generally on average are about 30% for 5-year survival rates.
And the cancer is more commonly referred to as RMS.
Yeah, we always abbreviate it as RMS. I think in the clinic they often refer to it as rhabdo. But in grants where we have to save on the number of letters, RMS works better for us.
So Kim, how did you get interested in this field of study?
Well, as part of like the university requirements, you have to do some sort of research or something. And so Dr. O'Brien just came to the school when it was about my time to start looking at research. And so I thought it was a great opportunity to gain some like awesome research experience. And then it just turned into a passion, and I really enjoy it.
Vittorio, what do you hope to learn through this research that'll carry you into a later life? And do you also want to continue on in the medical field?
I would like to do a PhD in cancer biology, and I hope that this experience will help me to learn more mechanisms about how cancer works and how we can treat it better in the future.
Why zebrafish?
Well, zebrafish, they have a really high reproductive rate, so they reproduce really fast. And we can inject DNA in their embryos, and their embryos are clear. So it's really easy to see if we inject right. And also the fish are clear as well, they're transparent.
So do you see cancerous tumors growing in the fish?
Yes, we can see that.
Yeah, we can, so the DNA that Vittorio was mentioning that we can inject into the embryos, that's how we perform genetic manipulations. And so you can do similar genetic manipulations in a dish of cells or with mice, but with a dish of cells, it's not a living organism, right? And with mice, it's a lot more complex, because as Vittorio, I think you mentioned, the zebrafish embryos develop outside of the mother, right? So you can do the genetic manipulations directly in the embryo without having to do any surgery. And so then we can genetically manipulate the fish so that their tumors fluoresce green. And then the fact that they're transparent helps us to visualize those tumors, as you were saying. We can see them as they're growing. Much easier.
So Kim, has the opportunity to participate in a research project like this and really get some hands-on laboratory experience in genetics, has it made you rethink where you want to go or what you want to do in life? Is this what you were thinking about when you entered school?
I definitely wasn't even considering research at all. But now that I've done the research, I think it's been like 2.5 years now. I really am thinking about it, and I'm really debating on getting my PhD and continuing with this kind of research.
Doctor, is research in RMS on the ground floor, or have we already made some great strides?
Oh, every type of cancer, there's been definite progress. And we focus particularly on RMS, but the genes that we study are upregulated or active in at least 15 other cancers. And so what we're hoping is, while we're focused on this particular cancer because we can model it in fish where they have a lot of muscle, we're hoping that what we learn can be applied to other cancers as well.
Now, you got some significant funding from the National Institutes of Health.
Yes.
$250,000.
Yeah, that's one way to say it.
What has that allowed you to do that maybe you weren't anticipating being able to do?
Yeah, so that funding came through in September, and so this is my 3rd year on the campus. And in the first 2 years, we were really just trying to get a zebrafish program up and running, you know, all the tanks, so that we had the fish alive and swimming and eating and figuring out the whole routine. But then once we got the research funding, we knew that we could really start asking these more intense questions a lot faster. So we had the injection equipment to be able to inject the DNA, but now we have funding that allows us to do all sorts of experiments to analyze the tumors as well, which would have taken a lot longer to get to.
So what's a typical day like in the lab? What are you doing when you get to the lab?
So every Monday at 3 p.m. we usually meet and we plan out the schedule for the week. And we are 6 students plus Dr. Boyan working, and so we actually have to fit like everyone's schedule. So we need to see when everyone is available. And so we plan that out, and then we meet during the week and we do what we decide on Mondays.
From the perspective of a student, what do you really gain from this hands-on experience in the lab?
Okay, so you need to study first of all, and what you learn in the book, it doesn't stay in your head very well. While if you do research, you put in practice what you're studying, and it remains in your head very, very clear, and you actually you learn way more and faster, I think.
Doctor, have you seen changes in your students by having them have this access?
Yeah, definitely. So when they work with me one-on-one, we get to know each other a lot better. And so I've seen a lot of changes, as you've mentioned, in career ideas and career directions. But we also try to bring these research experiences into the classroom and into the labs as well. So we use the zebrafish even without the research funding. I teach genetics and cell biology, and we've used the fish in both of those labs. So even students who aren't working with us, they get to have these hands-on or eye-opening experiences as well. And that's what's really fun, as Vittorio said, when you see a student or a student even says like, okay, I get it now, right? You were talking about this, I tried to do a problem based on this, but now I actually see it in action.
Why interest in this particular research? Did you have a family member that perhaps had this cancer?
No, so my PhD work was in breast cancer, pregnancy-associated breast cancer, and I did have a family member who had breast cancer. My grandmother passed away because of it. So that's what sort of got me interested in the cancer field. But as I mentioned, the genes that we're studying, they are also active in breast cancer and colon cancer and lung cancer, many common cancers. So when I knew I was more interested in a teaching career and I wanted to work on a smaller campus where I'd get to have these one-on-one opportunities, I needed to move from breast cancer, which is studied in rodent models, to something that was easier to do on a smaller campus. So then that's why I moved to the zebrafish organism as a model to study, and we can't study breast cancer in the fish. So we looked at other tumors that have the same gene family active in them, and that's where we realized that in zebrafish we can model the muscle cancer as well.
So what's the timeline for this? Do you know, is it going to be completed this year with the two of you working on it, or does this extend beyond? Any of you can answer where you see this going from this point.
Probably not. From lab experience, I learned that you need a lot of patience while doing research, and things will take longer than you expect. Sure.
I think we'll always have questions. So we might get answers to some of what we're asking, but those just bring new questions. And so there will always be experiences for us to be able to have questions about.
Well, this has been a fascinating discussion. All the best as you continue your research.
Thank
and
you.
thank you for being with us tonight.
Thank you for having us.
Thank you.